Hello again, and thanks so much for being here, and thanks to the group of organizers to having me here. This is really a very high-level meeting, and it's terrific to meet many old friends and make some new friends. So I'm going to talk a little bit about traumatic brain injury, which is an area that I've worked on for many years, and I think historically one of the early areas that Kai and Henrik's lab were very involved with. But let me go way back to the history of traumatic brain injury. I want to make the point that TBI is probably one of the oldest and most common maladies affecting humanity. In fact, even before we were humans, there's evidence of a South African Australopithecine individual, that 3 million year old skeleton. And this individual was murdered probably by another Australopithecine who hit him over the head with an antelope humerus. And we know that because the skull has a lethal skull fracture in it. It's very clear that one of the first things we did when we came down from the trees and discovered that we had an opposable thumb was to use that opposable thumb to grab a club and hit another humanoid or pre-human ancestor with it. This is in fact more than just anecdote. And there's evidence from physical anthropology, studies of ancient burial sites that have found that somewhere in the neighborhood of 15 to as much as 40% of all skulls, particularly male skulls, found at these ancient burial sites have evidence of skull fractures, either healed or lethal skull fractures. Our ancestors, it is hard to say, lived in a state of constant warfare with one another. They were constantly raiding the tribe of humans over in the next valley, bashing them on the head, stealing their women, stealing their crops. So any genetic variants, anything that would have helped us become more resilient from traumatic brain injury or would have helped us to repair and recover the brain would have been factors that would have been selected for by evolution. In the modern world, TBI is not quite as prevalent. We have had some civilization. On the other hand, it's still a very major clinical problem, and it's in fact somewhat an overlooked clinical problem. In the United States, there are approximately 3 million emergency room visits per year. The CDC study is somewhat outdated. The vast majority of those are considered mild, although that's very much a misnomer, because even these people who are evaluated in the emergency room, told they're going to be fine, sent home, somewhere in the neighborhood of 15 to 20% of those people are going to end up having disabilities that persist for months or in some cases even later. Approximately 300,000 per year get admitted to the hospital in a significant course of mortality and morbidity. And because this is primarily a disease of young people who survive and will live for many decades with the disabilities resulting from this injury, the prevalence of TBI-related disability is actually quite high. And the estimates are that approximately 2% of the US population is living with disabilities related from TBI. As a consequence of that very high social burden, there's been great effort over the last several decades to try and come up with neuroprotective drugs. And this has been an effort in traumatic brain injury, but it's an effort as well in stroke and MS and really many other areas of neurology. Now, unlike what we've just heard with stroke, in most other areas of neurology, and this is certainly true in TBI as well, this has been a successful effort in preclinical models in that there has been quite a few drugs and treatments that in preclinical models are effective in protecting the brain and in promoting neurological outcome. Unfortunately, and this is what I show here in orange, in every single case, when those therapies have been tried in well-done human clinical trials, they have failed to demonstrate any efficacy. This is a process that's still going on, and in blue are some drugs that are still being tested. On the other hand, they're being tested in the traditional paradigm of clinical trials, and I'm not terribly optimistic that we're going to have success there. So what is the problem? Why are we having this difficulty translating from well-done preclinical observations to failure in clinical trials? And I think there are probably many reasons, but one of the main reasons is that we have underestimated the complexity of the injury, and we have been assuming that the clinical trials are telling us more, are a better mimic of the human situation than they really are. So I'm going to make the case that we're going to have to understand the endophenotypes of traumatic brain injury, or if we're ever going to have any chance at all at developing effective therapies. So what is an endophenotype? Well, it's a $25 word, but the concept that it tries to get at is not particularly that profound. An endophenotype is an internal or intermediate phenotype that is closer to the biology of the underlying pathophysiology of the disease, whether it be genetic or environmental, and in most complex diseases, it's a mixture of genetic and environmental. Like, unlike a phenotype, which is something that is observable from the outside, and it's usually a categorical variable, meaning patient has the disease or they do not, an endophenotype, by definition, is a continuous variable. It's something that has to be measured with a biomarker, and that biomarker can be a physiologic, imaging, or a biochemical technique. So this is where we come in here. We're going to have to understand these biomarkers, understand how they, what they tell us about the activity of disease, understand what they tell us about the likely natural history, and then use therapies that will engage those biomarkers and move those biomarkers. So that all sounds pretty ethereal. Let's talk a little bit, just to give you an example of what endophenotypes of atherosclerosis are in coronary artery disease. So we know that a given patient with coronary artery disease, that's a phenotype, different patients may have different endophenotypes contributing to different degrees. Some patients may have hypertension, some patients may have hyperlipidemia, some may have vascular inflammation, some may have a mixture of each to varying degrees. And what's important here is that therapies, which we don't yet have for many neurologic diseases, but we do have them for coronary artery disease, are targeted not at the phenotype, but rather at the endophenotype. When a cardiologist evaluates a patient with coronary artery disease, he measures the blood pressure, he measures the cholesterol level, he measures the CRP level, and the therapies chosen are targeted at the particular biomarkers or the endophenotypes that are present in patients. So what are the endophenotypes of traumatic brain injury? Well, we don't necessarily know them all, but I think we know enough that there are several that are likely to be a factor. So inflammation is likely to play a role, diffuse axonal injury, and as well as diffuse vascular injury, and there are potentially many others as well. I'm going to talk about the remainder of my time. I'm going to talk about two endophenotypes of traumatic brain injury that my group in particular has been doing some work on over the last several years. And the first one of those is diffuse axonal injury. So diffuse axonal injury is an important cause of mortality and morbidity, and it is a consequence of the rotational acceleration and deceleration forces that occur upon the brain when it's involved with a fall or an assault, being hit on the head, or from a very rapid deceleration event, such as an automobile accident. Many years ago, my group, as well as many others, have been using imaging as a way to measure diffuse axonal injury. And a technique called diffusion tensor imaging has been very valuable in this method, and there are a number of ways that one can look at that. This is just an example using voxel-based morphometry, and what I'm trying to point out here is that you can detect within a couple of days of the accident, you can detect axonal disruption, but that axonal disruption actually goes on for many months after the injury, and when you scan the same patient again 6 months later, as you can see, the volume of abnormal white matter is much greater than the volume of abnormal white matter that was present immediately. So the initial injury damages the axon. Some of them recover, but many of them go on to decay over the next many years. Then part of the problem with DTI data is that you have so much of it, and that you have to use sophisticated, statistical methods to analyze it. And this is particularly an approach using discriminant component analysis. And what I want to make the point here is that the initial injury is not necessarily irreversible. In fact, in some patients, and this is what's in the green, over the first 6 months after injury, the DTI parameters appear to normalize, right? The movement of the vector is towards the upper left, meaning that they are again going to be overlapping with what healthy white matter should look like. On the other hand, other patients, the vector over the first 6 months moves rather to the upper right, and those are the patients that tend to have a less favorable recovery, as shown in this paper that we published several years ago. Now, more recently, and this is unpublished work that is being considered for publication in neurology, and this is work that Pashtun Shahim, who was a graduate student with Kai and Henrik back in Gothenburg, he's now working at the NIH with a group that I led for several years, has had the opportunity to measure neurofilament light in a group of subjects that were followed prospectively at the NIH Clinical Center. And these studies are very interesting in that they show that there's actually a pretty decent relationship between the neurofilament light levels in the serum and the imaging measures of axonal disruption measured by DTI, meaning either fractional anisotropy in the corpus callosum or a number of other measures as well. And in addition, in these TBI patients, we can see the same thing that has also been observed in multiple sclerosis, meaning that the degree of brain atrophy that occurs over time is associated with serum neurofilament light levels. So the serum neurofilament light levels in this TBI population is telling us something about the progressive brain atrophy resulting primarily from axonal degeneration and subsequent Wallerian degeneration. Now, another finding from that study is how long that process actually goes on. And in this slide, again from the same study, we want to show that even years after this traumatic brain injury, there is still progressive leakage of neurofilament light into the serum. And it's likely a consequence of persistent neuroinflammation that occurs after the brain is injured and goes on for a long time. And this is obviously much more the case in those folks that sustain what we would call a severe brain injury, meaning that they were in a coma for a period of time. But it's even the case in people who have sustained what we would classify as a moderate brain injury, meaning that they were not in a coma, but they required hospitalization, often for a couple of days. And to a lesser degree, even in people who would be classified as having a mild brain injury or a concussion, meaning that they were not even admitted to the hospital, but often had some disability. So the neurofilament light level decreases over time. It's very, very high soon after the injury, but that decrease is pretty slow, and particularly in the patients with severe TBI, there is a persistence of that elevation in neurofilament light even 3 to 5 years or more after their brain injury. So we believe neurofilament light has a great value as a potential biomarker to measure the endophenotype of traumatic axonal injury. The second endophenotype that I want to talk to you about is vascular injury. The brain is the most highly vascular organ in the body. The brain receives about 20% of the cardiac output, although it has only about 2% of the whole body weight. Because of the anatomy of the cerebral vasculature, it is particularly sensitive to the shearing and stretching forces that occur when mechanical energy impacts the head. And this is an example of a case of lethal TBI on the right, or rather on the left. This is a normal precapillary arteriole. You can see the vessel has a very smooth wall. On the other hand, after a traumatic brain injury, these vessels are stretched and sheared, and there is a thickening of the blood vessel wall, and this is the blood vessel that is not working properly. Now, that's the case in lethal TBI, but it's also common in much milder forms of TBI. This is a patient that we had the opportunity to study using a technique called susceptibility weighted imaging, and these black spots on this SWI image are an indication of these linear microhemorrhages that are the most common finding in CT negative mild TBI. So of the 3 million people that come to the emergency room with a mild TBI, pretty much they all get studied with a cranial CT scan. The CT scan is negative about 90% of the time. If you then take those 90% of cases that have a negative CT and one does MRI on all of them, the MRI picks up abnormalities in about 30%. Not a big surprise. We've known that MRI is far more sensitive than CT in just about every neurologic disease. And the most common type of MRI abnormality that one sees in these cases done in the emergency room is what I'm showing here, these SWI positive linear microhemorrhages. So vascular injury is a common cause. We've also known that it's a common problem as well in another condition that has not been the focus of our studies, but this is looking at professional athletes who've been exposed to dozens or perhaps hundreds of very mild head impacts. Likewise, the cardinal feature of the pathology there are these tau deposits that are primarily seen in the perivascular space. So several years ago, our group decided to try and use phosphodiesterase 5 inhibitors. These are drugs that can promote the action of nitric oxide and then promote cerebrovascular reactivity. We developed a imaging method by which we could study the normal cerebrovascular reactivity, and when we do that in patients who had had TBI, usually several years before, we find that there are persistent abnormalities, both in cerebral blood flow, we measure with arterial spin labeling, as well as with cerebrovascular reactivity, which is measured with this hypercapnia-Bolt technique. And then the exciting part is that we have been using sildenafil, which many of you may know better by the brand name of Viagra, in which we can use that to, at least in part, correct the cerebrovascular reactivity deficit that we see in these patients with TBI. And this is an example of where you see that. In the top row is a healthy control. In normal individuals, sildenafil does not potentiate cerebrovascular reactivity. On the other hand, in these two examples of patients who have had traumatic brain injuries, there is a potentiation of cerebrovascular reactivity, and it's not globally symmetric throughout the brain, but it's rather in those areas of the brain where there are particular lesions. So we're also interested, MRI is a very powerful tool to measure biomarkers, but as we've heard before, it's expensive, it's cumbersome, it's not well suited to repeated measures, so we're very interested in trying to develop blood biomarkers by which we can also assess this microvascular pathology, and this is a work that's pretty much in its infancy, but we're trying to match the blood biomarkers to the imaging measures, and here we have shown examples of two different types of vascular injury to the brain, being traumatic intracranial hemorrhages. This is what shows up on the CT scan, what often takes a lot of attention, and then the other example that I showed you earlier of these linear microbleeds. So we measured von Willebrand's factor and cellular fibronectin. These are proteins in the blood. Von Willebrand's factor is a product of endothelial cells. Cellular fibronectin is a product of parasites. And they, in fact, do differ depending on the particular imaging features of the particular type of vascular injury. And if you put the two together, they do add to the overall prognostic value over simple having clinical values as well. So in conclusion, I hope I've convinced you that the current classification schema that we have for traumatic brain injury are inadequate for the development of effective therapies, that we're going to have to understand the endophenotypes of TBI if we're ever going to have any chance of being successful for developing targeted therapies. We're going to have to combine imaging and molecular biomarkers and perhaps also physiologic biomarkers. I haven't talked so much about that. And finally, that microvascular dysfunction as well as axonal dysfunction is a potentially treatable mechanism of neurodegeneration, and that the future of precision medicine in TBI is going to require this understanding of endophenotypes. And this is just my group at Penn, and again, thanks very much for your attention.